Jennifer Duodna grew up in the small town of Hilo, Hawaii, and became very interested in the nature that was around her. She gained her interest in science from her environment and from her parents, who were both academics interested in evolution, astronomy, and geology. She gained her passion for biochemistry when she read a book by James Watson that was his personal account of the double helix DNA discovery. She also became passionate about science when she learned about how normal cells became cancerous. A struggle during her childhood was that she never felt like she belonged amongst her peers because they were all of Polynesian culture while she wasn't. Another struggle that she faced was being successful as a woman in science, a field that was traditionally male-dominated. These challenges did not stop her from achieving her dreams. She finished her undergraduate degree for chemistry at Pomona College in California, and then she went to Harvard University where she got her doctorate in biochemistry. She also had a lot of experience working in labs throughout her schooling, and in many of these labs she studied RNA with Nobel Prize winners. Because she grew up in Hawaii, she loved to explore the beautiful nature that was around her, and she loved to climb on volcanoes. She also has always loved to read books, and these passions that she had in life ultimately helped her form her love for science. One of the things that I admire about Jennifer Duodna is the importance she placed on family during her career. She went to work at Berkeley as a professor because she was able to be closer to her mother and extended family in Hawaii. I personally believe that family is the most important, so it's exciting to me to see when other people believe the same, and because of this, I identify with her. Another thing I admire about her is her perseverance, even when she may have faced struggles in a male-dominated field. Being a woman myself, it is encouraging to see women making huge impacts in the science field. Duodna is focused on biochemistry and specifically how RNA and ribozymes work. She was curious how RNA could give some hints as to how life originated when she originally started graduate school. She continued her research on the biological function of RNA after she graduated in a laboratory with another scientist who had just won the Nobel Prize for the discovery of RNA catalytic properties. The most recent paper that Jennifer Duodna is an author on is called Nontoxic Nanopore Electroporation for Effective Intracellular Delivery of Biological Macromolecules. This paper is about a method that Jennifer Duodna and other scientists came up with in order to more effectively deliver macromolecules into cells. This method they use is called simple nanopore electroporation, or nano-EP, which is a method of electroporation that is very localized, unlike the conventional method in which the entire cell membrane is electroporated. This method is significant because it allows for greater cell viability and greater efficiency for delivering macromolecules. This method also doesn't require any special buffers, cell manipulation, or expensive materials, so it is better in that sense too. In this experiment, Duodna and her team delivered nucleic acids, Cas9 single-guide ribonucleic proteins, and functional proteins into cells using this nano-EP method, and then they found a greater than 95% cell viability and an 80% delivery efficiency. Duodna's first breakthrough occurred when she was a doctoral student. Her research helped show that RNA helps catalyze the synthesis of proteins. She later was able to come up with a 3D structure of a ribozyme. One of her most well-known projects is CRISPR. Duodna came across these segments known as clustered, regularly interspaced, short palindromic repeats, otherwise known as CRISPR, and she studied how they worked because she believed her research could provide her with clues about how RNA molecules regulated genes in human cells. She then met another scientist named Emmanuel Charpentier, who introduced her to the enzyme Cas9. These scientists and many others worked together to figure out how Cas9 can snip out a piece of DNA using CRISPR RNA and track RNA as a guide. This CRISPR-Cas9 technology originated from the naturally occurring systems in bacteria, which provide them with adaptive immunity to viruses and plasmids. The technology that Duodna helped develop has allowed scientists to engineer genomic DNA in a more precise and easier way that could have so many benefits. However, Duodna lately has had to deal with more ethical problems that are arising due to her creation, as some scientists are wanting to use it to edit human embryos.